In this video, we'll be going through nucleic acids, which is a part of the biological molecules topic. Nucleic acids are found in all living organisms. It makes up their genetic material which codes for proteins that need to be made in organisms. If you refer back to the cell structure video, you will find this genetic material inside the nucleus. In your exam, it is important that you know the basic structure of nucleotides and are able to draw and label a nucleotide. So, as you can see in this diagram, it shows you a generic diagram of a nucleotide. You have a pentose sugar, nitrogenous base, and a phosphate group. There are two types of sugars that you need to be aware of. One is the ribose sugar, which is what makes up your RNA nucleotide. Then the deoxyribose sugar, which is found in DNA. As you can see, structurally, the deoxyribose sugar has one less oxygen. An easy way to remember this is looking at the first letter of the sugar, for example, R for ribose, then the R for RNA. In terms of the nitrogenous bases, there are two different groups to be aware of. One is purines and one is pyrimidines. Purines are a double ring structure, and there are two bases that are in this group, adenine and guanine. The next group is the pyrimidines, which are single ring structures, and there are three bases in this group, cytosine, thymine, uracil. The way I remember this is that adenine and guanine have two ends, hence both of these are double ring structures. In previous videos, we have spoken about condensation reactions to make phosphodiester bonds between nucleotides to form the polynucleotides. A phosphodiester bond forms between the pentose sugar and the phosphate group via a condensation reaction. The bonding alternates between the binding with either the third or fifth carbon on the pentose sugar. This eventually forms the sugar-phosphate backbone of RNA and DNA. In your exam, it is also important that you are able to remember the differences between RNA and DNA structures from comparison questions. In DNA, the sugar is deoxyribose, and in RNA, it is ribose. The bases in DNA are adenine, guanine, cytosine, and thymine, whereas in RNA, it has the same ones, but the thymine is replaced by a uracil instead. Finally, DNA is a double-stranded molecule, whereas RNA is only single-stranded. The structure of DNA was discovered in 1953 by Watson and Crick, using Rosalind Franklin's X-ray data to prove it. They found that DNA is a double-stranded helix, and the strands run anti-parallel to each other. One strand will be the 5 to 3 and the other 3 to 5. This is based on what I said previously about the phosphodiester bonding being based on the carbon number on the pentose sugar. They found that it had a sugar-phosphate backbone with the nitrogenous bases facing inwards. The nitrogenous bases line up with their complementary base pair in the opposing strand and form hydrogen bonds. So, adenine always pairs with thymine, and they form two hydrogen bonds between them. Then the cytosine always binds to guanine and can form three hydrogen bonds between them. The way that I remember this is that C and G are curvy letters, so there is more space to fit three bonds in them. In the next video, I'll be going through DNA replication, so be sure to give that a watch too. I hope you found this video useful, and please give this video a like. If you want to see more videos like this, then please subscribe.